Okay, it is 12 o'clock, so why don't we begin? I'd like to welcome you all to our spotlight lecture today. This lecture series is meant to put a spotlight on newer faculty who have come to Illinois or joined the IGB to help us learn more about the great work that they're doing and to help them get better connected into the IGB. Today's speaker, Professor Jake Sherkow, is a professor in the College of Law. Actually already is well on his way to being very integrated in the IGB, and we're delighted that he's speaking today in our Spotlight series. Jake is an affiliate of the Genomic Security and Privacy theme, one of the newer themes here at the IGB. Prior to joining us at Illinois, he held faculty position at NYU and visiting positions at Harvard, Columbia, Stanford, and University of Copenhagen. Professor Sherkow has a bachelor's degree from McGill University in molecular biology, a master's degree from Columbia University in biotechnology, and his jurisprudence degree from the University of Michigan. He is the co-author of a book and has published regularly in both law journals and science journals, with several published in Science magazine. He's an expert dealing with issues related to CRISPR, patenting genetic material, and other topics related to genomics. Professor Sherkow has had two prestigious clerkships in New York and in District of Columbia. He's also received numerous awards. He was selected by the National Academy of Medicine to be part of their Emerging Leaders in Health and Medicine group. He's won the Otto Walter Distinguished Writing Award from the NYU Law School and Leonardo da Vinci Research Grant from the Center for Protection of Intellectual Property. Jake serves on numerous advisory committees in science and law and the interface of science and law. He's an advisor he's on the advisory committee for the New York Genome Center, one of the prominent genome sequencing facilities in the country. Jake joined the Illinois faculty in 2020 during the pandemic. So as you can hear, he has great credentials. I'm not so sure about his timing, but we are really delighted that he has chosen to come to Illinois and to get involved with the IGB. And we're looking forward to his talk today on regulating genomic sequencing. Jake?
Thank you, Gene. Thanks to everyone for having me, for delivering this spotlight lecture. And really, if I may, I just wanted to extend a special thanks above all to you, Gene. You know, as you mentioned, I only joined the Illinois faculty last year in 2020. And I think you, aside from being the wonderful person and the faithful steward of the IGB that we all know you to be, were really kind and helpful and instrumental in my move here. So thanks. My research generally focuses on the law and regulation of advanced biotechnologies, which essentially means ways in which legal structures that currently exist affect the way bioscience is practiced. And today I'm going to focus on one aspect of this: how genomic sequencing is essentially regulated here in the United States. There are, in fact, and I think this is surprising to many scientists, if you stop and think about it, a bevy of regulations that govern genomic sequencing, not just who can conduct it, but how it is practiced. And so I'm going to begin by going through some of the basics in this area, and then suggest that actually most sequencing is regulated through one peculiar aspect: its intended use, rather than say who is ordering the sequencing or where the source material is coming from, or even the instrumentation that's ultimately involved, at least independent from use. I'm going to highlight two puzzles in this area, one from the clinical context. I will confess I'll be spending most of my time on the clinical context, and another on the human identification context that essentially arise from a regime that polices intended use. And I'll finish by offering just some observations of the future in a world in which genomics both is and also has a lot of difficulty being regulated in such a fashion. Some of these thoughts have come out of my genomics and law class that I've been teaching here at the College of Law. And so I would be remiss if I didn't thank my students for inspiring a lot of this discussion. And so some of what I'm sketching out here today with your input is actually also part of a project that I'm conducting, or at least about to start conducting at IGB under our GSP, our Genomic Security and Privacy theme, headed by Carl Gunter. And so thank you to Carl for your encouragement to think about issues like these. I'm going to speak for about 45 minutes before we take a Q&A. And so my goal this afternoon is to provide you all with a better understanding of essentially what my research entails, and for you to see some of the interdisciplinary scholarship in this area. But I also hope it gets you excited to talk about genomics and the law as an area of study, and to impress upon you non-lawyers in this audience, which seems like is the majority of you, or at least I would hope, about how your scientific expertise is needed and valuable in the field to inform scientific law and policy. So during the past 20 years, there has been an explosion in genomic sequencing, a statement that I think to everybody here on this Zoom is like so obvious that maybe it's meaningless, right? You have all seen this graph. It must be presented in every single talk as a demonstration of this revolution in genomics, where we see the price per sequenced base falling many orders of magnitude, at least since the 2003 completion of the Human Genome Project. I think this graph is misleadingly incomplete. It's worth pointing out that it measures just one aspect of genomic sequencing, and that is cost per base, right? That is essentially what the y-axis tells us here, and nothing more. It does not measure the astronomical growth in other aspects of sequencing, namely the rise in the volume of sequencing that is conducted, an explosion as to the source of material that is used, and the resulting increase in the diversity of uses of genomic material once sequenced. To get a better appreciation for that, take, for example, this single chart from GenBank. This is the National Center for Biotechnology Information's Open Access Data Initiative. Simply looking at this one database, simply looking at this data from GenBank, that is, you can see that the number of genome bases sequenced in total since 2003 has gone from about 0 to well over 10 trillion, an increase of 13 orders of magnitude in not even 2 decades. There has also been an explosion as to the source of genomes being sequenced. So not to slight bacteria or archaea, please forgive me, Professor Woese, but the first full eukaryotic organism to undergo whole genome sequencing was in fact baker's yeast. This is our favorite friend, Saccharomyces cerevisiae, back in 1996. Today, GenBank has data from about 300,000 eukaryotic organisms, hundreds of which, if not thousands, I will confess to you, I haven't bothered completely counting, are complete genomes. And that's only GenBank. Beyond all this, and this is what I think is significant for my talk, there has also been an explosion in the diversity of uses for genomic sequencing. Today, whole genome sequencing is not a mere curiosity, but is routinely used for a variety of purposes, including medical diagnostics, clinical treatments, human subjects research, non-human applied research, such as that for conservation biology and ecology, human identification, and of course, fun and profit, as we see in the direct-to-consumer genomics industry. More on that later in my talk. So when we're thinking about the genomic revolution and how it brushes up against the law, I think it's important to keep in mind that the revolution has not been one simply of price, as that comparison to Moore's chart so simply dictates, but also one of scope, scale, and importantly, use. So what does this revolution in genomics mean for the law? Like, why am I here, right? Well, the explosion in the variety of uses of genomic sequencing makes it such that genomics now has the potential to touch upon a wide variety of areas of human activity that are otherwise regulated in a variety of ways. And as the scope and scale of genomics are increasingly expanding in those areas, the instances of regulation increase. For but one example, which I'll flush out more in a bit in some detail, as genomics increasingly becomes part of bedside medical practice, regulations surrounding such standards of care are increasingly likely to affect how physicians go about ordering and interpreting genomic sequences, how they do so, essentially what they order, and what this means for patients, and of course, their insurers, who have a strong vested interest in managing just how this is done. Before diving into these areas, I want to try to separate two aspects of law for purposes of this talk. And that's this nebulous, if not entirely ethereal line between the difference of law from regulation. There is, of course, no clear-cut definition between the two words. Is the law pertaining to the Food and Drug Administration, that is the FDA, a law because it's codified in a congressional statute? Or is it a regulation because it concerns a federal agency? Reasonable minds could, and they do, trust me, strenuously disagree as to whether this is a meaningful distinction or not. Rather, what I'm trying to get at instead is a sense of regulation insofar as that word applies to legal requirements that either affect public actors, like the arms of government, or are mediated by public actors, like federal and state agencies. This is different, I think, than other aspects of law and genomics that are probably on your mind. Many of you who play around with genomic data sets may have a narrower conception of these issues. You may think, maybe there are some things to do with data privacy, maybe there's some issues pertaining to intellectual property, maybe that's about it, right? These are obviously important topics, and given that they are a principal aspect of my research, I certainly don't want to suggest otherwise. But thinking about them solely in these terms, essentially privacy, intellectual property, which largely revolve around what legal scholars call private law, is, I think, a bit narrow in scope. It essentially concerns only two areas of law, property and contracts, which I really won't be touching on today. Just as the uses for genomics has expanded, so too I would like to suggest we should expand our thinking about which aspects of the law the field touches on in the first instance, right? All right, enough with these abstractions. Let's make this more concrete. How is genomic sequencing regulated in the United States? I'd like to suggest to you, I guess the thesis for my talk, if there is one, is that it's by the sequence's intended use. And to explore that a bit, let's think of a hypothetical to show you this in operation. Imagine, for example, a patient goes to their physician, and the physician orders a whole genome sequence of the patient. Which laws, if any, cover this interaction, and how? The answer is, as it is for all good legal questions, it depends, right? Namely, it depends on what the physician intends to do with the sequence, i.e. the sequence's intended use. Why is the physician ordering this sequence in the first instance? If the physician is using the sequence to diagnose or treat the patient, for example, then a wide variety of regulations begin to apply. Regulations pertaining to medical practice, regulations pertaining to certifying the laboratory conducting the sequencing, regulations pertaining to patient privacy, and even regulations pertaining to the sequencing instrumentation itself. If the physician is using the sequencing to conduct research, by contrast, then a different set of regulations apply, namely those concerning protections of human subjects and research, found most significantly in what's known as the common rule, the cross-agency federal regulation setting forth requirements regarding informed consent, as well as the makeup of institutional review boards and their ambit, and potentially, FDA's investigational device exemption requirement, or IDE regulation. But let's say the physician is using the sequence for yet another purpose. Perhaps it's part of an employer voluntary wellness program, Or perhaps it is under the direction of a particularly savvy life insurance underwriter. Or perhaps the physician is using it to establish or disestablish paternity at the behest of a patient, or to identify the patient as a match, as a donor for organ transplantation. Then a whole host of different regulations apply. This hypothetical, I think, demonstrates that the regulation of genomic sequencing is ultimately about what the intended use of the sequence is. Me simply providing you a hypothetical that says a physician orders a whole genome sequence is simply not enough information to assess what the regulations are. Instead, it's ultimately the intended use that governs how such sequencing is regulated in the first instance, right? To better understand this all, let's explore some of the regulations I listed in my hypothetical before to see how they in fact affect the makeup of genomic sequencing. So let's begin with the scenario in which the physician is ordering a patient's genomic sequence for the purpose of diagnosing or treating the patient. In that scenario, regulation affects both the sequencing itself and the delivery of sequence information, both to the physician and to the patient in several ways. Perhaps first, or at least the first point at which regulations governing sequencing conducted for a clinical purpose is the laboratory in which such sequencing occurs. The Clinical Laboratory Improvement Amendments, or CLIA for short, require laboratories that return clinical results to be certified by the Centers for Medicare and Medicaid Services, also known as CMS. The certification is designed to ensure essentially one thing and one thing only, and that is the analytic accuracy of the lab's tests and the way that they are routinely performed at that particular lab. Namely, what we want to know is, are the labs accurately measuring and reporting what they claim to be measuring and reporting? In the genomics context, this means variant calls. Are CLIA genomics labs correctly identifying the nucleotide bases in the sequence genomes? That is, are they getting the ACs, Ts, and Gs called correctly? This may sound at first like an empty requirement, right? You may think, well, if that's regulation, that ain't much. But in reality, in order to get to that point, you may be astonished to find that CMS promulgates a long list of regulations pertaining to test quality for genomics, and then further interprets those in a series of guidances known as the interpretive guidelines that add specific details regarding how genomic testing is to be conducted at CLIA certified labs. For example, the interpretive guidelines specify the controls that are to be used in certain genomic tests, separating HLA tests from non-HLA ones, for example, the primers and the probes used to conduct certain sequencing, regulations pertaining to next generation sequencing specifically, as opposed to that more of the Sanger type, DNA isolation procedures depending upon the reference material, and so on. That is, government regulations control the mechanics of how sequencing is conducted at CLIA labs, that is, to the extent they want to remain certified. Beyond this, CLIA establishes minimum reports for labs, including a requirement that any report generated by a CLIA lab that is returned to a patient list the precise mutation identified, if any, for any target gene sought, and that patients can, upon request, see the original variant call file or VCF file that's associated with their test. Don't encrypt your BAM files too much such that you can't return them to patients. This is a clear regulation. Packaging these together, this means that there are in fact regulations that determine how sequencing is actually conducted, down to like which controls a laboratory can use to validate its test and file formats that sequencing information needs to be stored in. And indeed, there are entire service companies built around managing and documenting CLIA services for genomics, including one called Collaborate, for some reason spelled only with one L, and another ACGT Inc., which I have to say is a great name for a genomics CLIA compliant testing company that make themselves the regulatory compliance professionals of the genomic world. This story, I think, runs pretty contrary to popular assertions that what we're living in now is like the Wild West of genomics, where essentially anyone can open up their own genomic sequencing lab, especially as the cost of desktop sequencers continues to fall and Illumina's patents begin to expire. Instead, I think it makes clinical genomics look something more like a regulated industry, more like the way that, say, we build cars or airplanes than the way that we govern people building apps on the weekend. Second, aside from this, all of this applies to genomic instrumentation itself. Virtually all sequencing labs of which I am aware are not using some custom-based sequencing equipment, but instead off-the-shelf standard sequencing instrumentation, typically from Illumina, which, note, seems to operate a monopoly in this area, and why that is not otherwise considered an antitrust violation is a puzzle that scholars will think about for a long, long time. And so this sequencing instrumentation for use in clinical labs is also regulated. Generally speaking, instruments that are, quote, intended for use in the diagnosis of disease or other conditions, in the cure, mitigation, treatment, or prevention of disease in man are medical devices and subsequently under FDA's purview. I think to the surprise of a few scientists, this actually means that some of Illumina's products are quote unquote medical devices according to FDA law. And these include Illumina's NovaSeq, MySeq DX, and NextSeq DX systems, among others. This also includes Illumina's universal kit reagents, which recently received a Class 1 designation from FDA. By recently, I mean a few years ago, but still. All, after petitioning by Illumina, have received some form of formal clearance from FDA for use in clinical applications. And importantly, only those instruments that have been cleared by FDA can be used for clinical purposes in a CLIA certified lab. No, just because you're operating a CLIA certified lab does not necessarily mean you could run an Oxford nanopore machine against it, against a sample that you otherwise want to sequence. Thus, although we now have an explosion of sequencing instrumentation and modalities, some of which, like drop-seq methods, are relatively advanced and pretty cool technology, they cannot be used to return clinical results to patients, at least according to the CLIA guidelines that we otherwise have. So here too, right, contrary to the perception that genomics is an area that is, quote, free to innovate, a large part of the business of sequencing instrumentation is actually the selling of regulated products. One last area in which this seems to be significant is storage. Storing patient data in the days of genetics, right, single genetic tests, single gene sequencing, right, was, if not at least conceptually simple, feasible in individual laboratories. Patients' records could simply be kept with the labs that conducted the tests themselves. But in age of genomics, this becomes almost impossible. Whole genome sequences are often housed in relatively massive files. For those of you on this call that work with BAM files, you are intimately aware of this. These files at 30x coverage, right, fairly standard, if not clinical grade, whole genome sequence coverage, can be upwards of 80 gigabytes per whole genome sequence of a human. A mere 10 of those wipes out a standard 1 terabyte hard drive. If you are, say, the Mayo Clinic, which is embarking on a project to provide whole genome sequences to 100,000 of its patients, that is conservatively 8,000 terabytes. Where do you put all of this information? And the answer lately is cloud computing. And there is too an entire cottage industry that is devoted to cloud storage just for whole genome sequences. Some of these security issues notwithstanding, which itself is an entire subject in the literature, storing such sensitive health information in the cloud by third party raises major HIPAA issues concerning data portability, preservation, and access to patients. And as a lawyer, and because I'm obligated to mention it, who is responsible in the event of a breach or unauthorized access of the same? These bring in a host of regulations, including levels and standards of encryption that is mandated by HIPAA's data security requirements in an umbrella set of regulations concerning personal health information. And I just need to pause here and just note that Jan Joly's work at McGill University essentially instrumental in this regard. This is complex because a patient may wish to transmit some data, but not others, in an effort to obtain medical treatment, or even, for that matter, to share it among providers that are spread across different health systems. How can one do so securely? Or perhaps if you want to ask an even more nuanced question, how do we get patients to trust that providers will in fact do so securely? If that last question interests you, that's wonderful news, because this is in fact part of a project that I'm working on with Carl Gunter here at IGB in conjunction with Alex Shakovitz and Steven Schneider. We're a motley crew, a computer scientist, a lawyer, and a political scientist, and a sociologist. And we're trying to figure out ways of engineering systems that allow the partial, secure transmission of genomic information in a trusted environment, and to do so in a way that's both legal, that people will actually use, right? But we're also under no illusions, or at least I don't want to speak for everyone, at least I'm under no illusion. What makes all of this necessary, what makes me, the lawyer, necessary in all of this, is that this is largely a regulated field, right? Amazingly, this entire regulatory apparatus governing labs, sequencers, data storage, simply does not apply in the research context, right? If you're conducting research on genomic data, that is, if you're not engaging in sequencing for a clinical purpose, there is no need to use a CLIA lab. If you're not intending to diagnose a condition, then the sequencing apparatus is unlikely to be an in vitro medical device. If what you're producing is not health information, then there is no HIPAA concern to worry about. Rather, if genomic sequencing is being conducted for research purposes only, then a different set of regulations apply. Rather than walking through those as I did with clinical regulations regarding genomics, I want to explore instead how this is a fuzzy line. This is one of the puzzles that I mentioned earlier. What's intended use exactly? I'll let you noodle over that. Is it intent according to a reasonable person, one of lawyers' favorite metrics? Is it actual subjective intent, that is what is going on in the brain of the particular researcher ordering the sequencing? Is it objective intent according to the actions of a researcher? Intent is always difficult to divine. To paint this portrait of intent in sequencing, let's consider another hypothetical, right? That's essentially the picture that you see here. A genomic researcher who's also a rare disease clinician gets a patient referral. A pediatric patient is on a diagnostic odyssey. The patient seems to have a rapidly developing neurodegenerative disorder, but identifying it, including doing so through whole exome sequencing, has uncovered nothing. Our researcher is interested in, among other things, the non-coding region of the genome's effect on rare diseases, and so orders not a whole exome sequence for our patient, but a whole genome one, and does so with pretty deep coverage. Let's say, I don't know, 200x, right? Lo and behold, our researcher discovers a mutation in the intron of a single copy of a gene, which further research identifies as yielding a splicing defect in that particular gene. This particular splicing defect makes a truncated protein in a gene known as CLN7 that has a role in neurodevelopment. The mutation, though, is a single base, non-coding de novo mutation that, at least as up to this point, was totally unknown to the literature and humanity in general. But the research doesn't end there. The researchers then use that genomic information to develop an antisense oligonucleotide to target that area of the patient's genome in an effort to repair the splicing defects. They print off this nucleotide, that is essentially, they get it synthesized, they create it into a pharmaceutical formulation, and they deliver it intrathecally, that is into the spinal fluid of this particular patient in an effort to treat them. This may seem like an all too powerful story for genomics, but it is in fact not a hypothetical at all. This is the story of Professor Timothy Yu and milasen, the world's first truly N-of-1 therapy that also happened to be genomically engineered as a result of sequencing. It was called milasen because it was specific to a single person Mila Makovic, who you see below here, who has since tragically died. The creation of milasen challenges a lot of paradigms about medicine and genomics, but also regulation. The publication of this research also merited an op-ed in the New England Journal of Medicine by then directors of FDA Janet Woodcock and Peter Marks, who called it a new, quote, paradigm in drug development. But back to the original choice to resequence the patient. Was it intended for diagnosis, or was it intended for treatment? On one hand, it was used to investigate the source of an unknown mutation as a demonstration of the need not just for whole exome, but whole genome sequencing, and deep sequencing at that, for patients in the event of a de novo mutation. There was an observation, there was a hypothesis, there was a test, and there was a conclusion that was otherwise drawn from the results. That seems like paradigmatic research and human subject research at that. But on the other hand, the sequence information was ultimately used not just to diagnose, but in fact to treat the particular patient and even to create an investigational product itself. That seems like clinical use. In addition, it seems to be the case that the intent changed as the results came in. What started off as a research question became a clinical one as the information about the mutation became apparent. So what then? Answering these questions may require some philosophical gymnastics, or at a minimum, different paradigms about how one goes about assessing intended use and whether or not the intent could change as results of sequencing come in. This case may represent a paradigmatic shift, not just in the regulation of drug development, per Woodcock and Marks, of course, but also with respect to genomic sequencing itself and how to govern its ordering. Answering these questions about the research versus clinical divide has some significant practical implications. This is not just legal philosophy. If they remain to be seen as research, then the human subjects protection paradigms otherwise in play in the milasen case are essentially the governing body of regulatory law. No CLIA certified labs, for example, are needed to conduct this particular work. But if they become to be seen as a larger diagnostic or treatment pathway, then all of the regulations that I described earlier about CLIA certifications, about HIPAA, about the way in vitro devices are regulated by FDA, all of those begin to kick in. And it's not entirely clear that those regulations, especially when taken piecemeal, are up to the task of something like this. Further, making them so seems to cabin the choices that researchers will ultimately have in which sequencing protocols they feel comfortable of using and which standards they can employ in validating their sequences. For truly de novo mutations, especially ones in unexpected areas of the genome, it's not entirely clear that the suite of regulatory paradigms we have allows researchers the flexibility needed to design sequencing protocols that are up to snuff. Perhaps the de novo mutation that we're dealing with in a given patient relates to a transcription factor, but validating the transcription factor's clinical effect in a given tissue may require conducting some drop-seq variant, right? Controls for many of these drop-seq protocols are not necessarily standardized. Many of them are home brewed. And so what exactly can a CLIA lab do to establish standards for analytic validity in that context, especially on instrumentation not otherwise authorized as an in vitro device? To be clear, this is surely the path that we are headed down. It's unlikely, as successes mount, that we can continue to simply just call this work research and sweep it under the human subjects research protection rug. At some point, it just becomes regulated genomic medical practice. What do we do after that? Beyond all of this, I hope this highlights too some of the difficulties in regulating not just use at the time of sequencing, which is what I've been talking about, but actually use after an initial round of sequencing has already been conducted and you have a genome sequence in hand. This is, of course, the paradigm of secondary use. Imagine instead that an individual undergoes deep whole genome sequencing, and this information is otherwise stored online in the cloud, as mentioned earlier. At the moment, the individual is otherwise perfectly healthy. There is nothing, in other words, to diagnose or to treat, nor is there anything actually being researched per se. It's simply informational content about this person's biologic makeup. This would mean that neither CLIA nor FDA's IVD regulations immediately seem to apply. And it would also suggest that any regulations pertaining to human subjects research wouldn't be apparent either. Later, however, the individual becomes sick and a genetic cause is suspected. Can physicians and researchers later dive back into the patient's genome to potentially diagnose and treat the patient, or at a minimum, to research its causes? These questions surrounding secondary use of genomic information have plagued researchers for decades, and there is both deep and broad literature in this area, as there has been for at least 30 years. But these are more salient today, especially where whole genome sequences are becoming increasingly common, stored in third-party servers, and available to patients for release, in addition to being ordered for purposes that are not immediately diagnostic or research-based. In a world where genomic sequencing is ubiquitous and where the data that is produced from it is permanent, regulating genomic sequencing seems, if not ultimately impossible, at least momentarily fruitless. All right, now for something completely different. I've focused on the complexities that arise in the context of the clinic, but medicine is not the only human activity where genomics is important. Genomics is also useful, and indeed pretty powerful, as you likely know, for purposes of identification. This is, of course, most salient in the criminal context, where a primary objective, and here genomics is obviously a gift, if nothing else, to investigators. It allows law enforcement to use sequencing to match DNA samples found at a crime scene to those that they otherwise have, either from another suspect or from a database of collected genomic information. The problem, of course, is getting a DNA sample from a suspect in the first instance. And this, I'd like to suggest, is a regulated practice. Indeed, obtaining DNA directly from a subject, at least against their consent, is regulated not by some agency interpretation of some statute, but in amendment to the US Constitution, that is the Fourth Amendment. The Fourth Amendment protects against unreasonable searches and seizures, except where there's a warrant issued on a finding of probable cause. For genomics, this means forcible buccal swabbing, right? So when can it be done, right? Well, obviously, if the police have a warrant, that is, if they have probable cause to a particular suspect of a particular crime, at least incident to arrest, this is otherwise permitted under the Fourth Amendment. But beyond that, almost every state in the union has laws for allowing buccal swabs if you've been arrested, that is merely arrested, for a crime of violence or a probable felony, right? But many too have regulations concerning who is authorized to sequence such samples and when such samples must be destroyed. In 2013, the Supreme Court upheld this interpretation of the Fourth Amendment in Maryland versus King, and this is essentially the courtroom sketch of those oral arguments, or at least the actual delivery of the opinion that you otherwise see here. And suffice it to say, whatever sequencing is done need not be done in a CLIA lab or stored in any way that meets HIPAA protection. It simply needs to be authorized by the state for use in identifying a criminal suspect. Again, it is this use of genomic sequencing that seems to be the linchpin to regulate it. But there are more ways of getting at genomic information. As genomics has become more commonplace, law enforcement is now turning to direct-to-consumer genomic ancestry databases to narrow down suspects in cold cases, especially as they relate to how those suspects fit in to familial branches. This happened recently in identifying, after decades of dead-end leads, the Golden State Killer, the serial murderer and rapist in California in the 1970s and the 1980s. There, researchers resequenced samples left at the killer's crime scenes and compared them with those on GED Match, a free online whole genome DNA database used mainly for ancestry purposes. In doing so, they narrowed down the suspect to a broad family tree of individuals who had deposited their information there. By taking voluntary samples from those individuals and homing in on one suspect, this is Joseph James D'Angelo, they were then able to obtain DNA unwittingly left behind by D'Angelo on a coffee cup and matched it to that found at the crime scene. This use of genomics for, let's call it surreptitious identification in the criminal context, and I just want to be clear, this is a term of art. By surreptitious here, I simply mean without a warrant or probable cause to suspect this particular suspect, has had many scholars lamenting that we live in an age of a genetic panopticon without sufficient Fourth Amendment protections. But the primary legal complaint is that because users are voluntarily depositing this information online, and because it can be used to identify family members who did not otherwise consent to it, there is little the law can do about it. And frankly, under what's called the third-party doctrine of Fourth Amendment jurisprudence, they're right. Willingly turning over information to a third party generally means the individual has lost a reasonable expectation of privacy. And so depositing that information online, just like putting your information on Facebook, means that law enforcement can comb through it just as much as, say, your third cousin who is really interested in ancestry and seems to message you all the time. Here too, this seems like genomic for uses in criminal identification is an unregulated field. Rather, it seems like it is governed by the services providing this information with use by members of the public with or without badges, as a Fourth Amendment otherwise dictates. This belies, I have to admit, some recent attempts to regulate this area. In the federal context, notably, the Department of Justice put out a 2019 memorandum regulating its own use of genomic genealogy services in conducting its investigations. There, DOJ limited its use of such technologies to only, quote, unsolved violent crimes, or crimes that present in a substantial and ongoing threat to public safety or national security. Much like CMS's regulation of clinical genomic instrumentation, or FDA's regulation of sequencers under its IVD authority, DOJ has made an otherwise open practice subject to regulatory strictures. Several states have similarly attempted to do so. And aside from uses in crime, law enforcement is similarly investigating using genomics as part of identification process in immigration proceedings, made all the more sadly salient due to the previous presidential administration's zero-tolerance border policy that separated migrant children from their family members. This is controversial for a variety of reasons, but it's simply worth noting again that the use of genomic sequencing governs which policies apply. All right. So I've presented a span of what regulating genomic sequencing looks like today, but what about tomorrow, right? And I think there are three things worth considering. First, an expansion in the uses of genomic technology, right? Obviously, secondary use is going to become increasingly problematic as people get genomic sequencing for a variety of non-clinic and non-research purposes and put them online. This seems to suggest that there's a bunch of opportunities out there for regulatory arbitrage. Why go through the process of putting in through an insurance claimer sequencing of a patient's genome rather than asking them, have you already done this through Nebula Genomics, for example, yourself, and taking the information from there, even though none of that is otherwise conducted in a CLIA facility, right? In other words, to the extent that patients or subjects are otherwise doing this themselves, or doing it for a non-clinical purpose and want to eventually use it for a clinical purpose, this presents the opportunity for regulatory arbitrage. Second, I think it is fair to say that the uses for genomics are going to become increasingly routine for medical practice, and to the extent that such testing is in fact ordered in the clinical context, I think one of the things that we're going to see moving forward is that it is largely going to be insurance driven, right? Why pay hundreds, if not thousands of dollars for a 30-gene panel test for cancer for someone when you could simply pay for whole genome sequencing and have it done once and have it done forever? This also raises a bunch of questions regarding the portability of such information as people change insurers, which may happen in contexts that have nothing to do with health, such as when, for example, people change jobs, right? One of the fears that we may have in this particular area is to the extent that insurance becomes increasingly involved, that we may be otherwise worried about capture. That is, worries about which genomic sequencing gets to be conducted or whether and to what extent it is otherwise portable from one clinical context to another is going to be dictated by them, right? As opposed to ways that make sense either from a privacy perspective or from a clinical one. Lastly, I think it is fair to say one of the things that we may be worried about is regulatory accretion. I think there's fewer opportunities for comprehensive change as the number of arms and agencies that are involved in regulating genomics seems to increase, and the number of regulations that we have goes up as well. I think this makes regulating new applications particularly challenging. One of the things that is being discussed lately, for example, is rather than conducting panel gene sequencing of newborns, this is the heel prick blood tests that many do in certain jurisdictions. What exactly are we going to do if newborn screening is instead whole genome sequencing? Who owns the data? How do we access it? Who otherwise pays for it? Can it be used in the clinical context, and by whom, and under what circumstances? All of these questions, we have a regulatory apparatus that is already in place that is going to function in this area, even if it is not otherwise designed to do so. So I wanted to finish with a quote from a completely and utterly different context, that is tax law, to suggest just how complex things have gotten and what some of the duties researchers in this area otherwise have. Here is what my favorite Supreme Court Justice Jackson happened to say about tax policy. No other branch of the law touches human activities at so many points. It can never be made simple, but we can try to avoid making it needlessly complex. I think that's one of the things that people working in this area can otherwise endeavor to try. I hope that we can do so. So with that, I'm more than happy to take questions. Thanks so much for having me today.
Great. Thank you very much for that great talk, Jake.
Thanks.
So we're now open to questions. Feel free to put them in the chat if you'd like me to read them, or if you have a question, you can raise your hand and I can call on you. Let's see. I'll start, Jake, with a question. You touched upon the issue of medical testing and where genomics fits into this. It reminds me that our very first Genomics for Judges course, we had Gina Colada speak. This is now almost 10 years ago. And she painted the scenario that you touched on and then approached it from the medical ethics and HIPAA perspective. So she spoke about the fact, mind you, this is 10 years ago, that at the time, many tests were single gene or a few gene panels, as you referred to them. But it is becoming, obviously already back then and now for sure, easier, simpler to just sequence the whole genome. So the question she posed was, what if someone, a medical practitioner, orders up a test for gene X as a clinical diagnostic test for disease X and finds a mutation in gene Y that predisposes or causes disease Y? Is the practitioner allowed to share that information under HIPAA, and what are the conflicts related to medical ethics and regulation? So has that come up? Is that an issue as we've gone, as you said, more and more to genome sequencing? Love to get your perspectives on that.
Yeah, so this essentially concerns what some scholars have called the return of incidental findings, right? You're essentially out there looking for one particular genetic condition and you come across another one. And the circumstances under which you deliver that information to the patient, it's pretty nuanced, right? I think at least as of right now, some of the recommendations that have been floating around, I think it's either by ACMG or it's by ACOG, right? Is that essentially such conditions should not be returned to patients unless they've otherwise opted into this particular practice, if they're not going to be clinically actionable, which may be particularly problematic if you stop and think about it for a second, because there's a whole host of research being conducted on a lot of the direct-to-consumer genetic tests that even if consumers get information that's not otherwise clinically actionable, they still otherwise want to know. Like they want to know something about themselves, they want the ability to plan their affairs or not. And there are some conditions that require medical intervention, even though they're not otherwise clinically actionable because they seem to be fatal, right? So for example, a patient who otherwise comes in and they want to get a genomic sequencing because they feel like they have a predisposition to a particular type of early onset cancer, but it otherwise is revealed that they have a de novo mutation in the Huntington's gene, right? Like what exactly do you tell them? Right? On one hand, it's not quote unquote clinically actionable, right? There's nothing you could do for them except palliative care, essentially. On the other hand, knowing this information allows them to plan their affairs in such a way that we would think that not providing that information to them would be like ethically dubious. Ronald Green at Harvard has some really wonderful work about just this, about this fear that returning non-clinically actionable information to patients is always going to be bad. And so I think there's some actually empirical evidence to suggest otherwise. I think in a more practical perspective, is the route that a lot of the sequencing is going these days is through what are called broad consent platforms. That is, everyone is aware of some of these pitfalls now, and a lot of them can be, lawyers would say, contracted around by simply obtaining broad consent from patients and subjects. So just give them an informed consent waiver that says, I consent to everything, right? At that point, right, they've essentially opted in. And so at least that legal stricture with respect to their consent, especially as that's, otherwise mandated under the common rule, that's at least gone. We can talk about whether this is ethically sensible or not. We can talk about whether people appreciate the differences between a narrow consent form and a broad one. But still, that does seem to be one of the paths that are otherwise going on now. And Jean-Tine Loonshop's work is particularly helpful here too.
Great, thank you. Let's see, we have a question from Carl. Carl, do you want to state your question?
Sure. So we're going to be looking at legal issues for many different types of omics coming up over as they become more widespread. I wondered how much of a guide do you think you'll get from where we are now with respect to the genome? In other words, you're going to get good, good analogies, or do you think it'll probably be completely different for omics of other kinds? In particular, I wondered if you could talk about epigenetic clocks.
Yeah, I think a lot of this stuff centers on a tricky distinction or point regarding the, let's call it the temporal scope of the somatic genome relative to some of these other omics things. And so what do I mean by that? I mean, the somatic genome's a somatic genome's a somatic genome. A lot of our genomic exceptionalism that is built into the law and that we think about it centers on the fact that it's immutable, it's unchangeable, it's with us forever. But a lot of the omics work is transitory, right? If you're doing a bunch of tissue profiling on, which proteins are expressed in a particular tissue, well, that's only true today. That may not necessarily be true tomorrow. And there may in fact be a lot of environmental factors for which people have control over that go into doing just that. I think one of the interesting applications of this, or one of the interesting principles, if you kind of want to use this as like a philosophical razor, is whether or not your genomic exceptionalism extends to your microbiome. I think either Carl or it was you, Gene, that described it as this, right? Like your genome is who you are, but your microbiome is just who you are today, right? And so given that, right, that tends to, I think, kind of cut away from a lot of the genomic exceptionalism stuff that we're otherwise concerned about. And I think also by virtue of that may even kind of tamp down on some of the privacy concerns that we would otherwise be fixated on. So I think that there are some lessons to be learned about like practical, who is going to be allowed to conduct this work and where do we store the information and kind of that kind of stuff. But I think thinking about the attachment of like personhood to some of this information, I think is not going to be super informative. But we'll find out, right? I don't know.
Thank you.
Okay, we have a question from Chris Fields, who is one of the directors in our Carver Biotechnology Center. So it's a very practical question. If one is a provider of research-specific genomic services, say, for example, a certain biotechnology center that provides sequencing and analysis services that's not CLIA or HIPAA compliant, what should they be aware of when accepting samples from PIs? Is being proactive enough, namely making all PIs aware that we do not accept samples that might fall under CLIA HIPAA? Is that sufficient, or is there a further responsibility on the part of the directors?
Yeah, this is a difficult question that at least centers on whether or not one is a covered entity or a business associate of a medical provider under HIPAA, and that is going to entirely depend on the nature of the relationship between researchers who are giving their samples that may otherwise surreptitiously be for clinical purposes. I think that's what you're otherwise worried about, right? Versus ones that are not. And with respect to what happens here at University of Illinois, like I would imagine that that would be even more complex because essentially the university is a state-run agency, right? And otherwise has a bunch of, that tends to somewhat complexify some of the CLIA requirements that otherwise exist. So I think it's going to depend on like the granular circumstances of obtaining some of these samples, right? In terms of assessing, like, are you unwittingly delivering clinical information, whether or not liability is going to attach to that, right?
Yeah, that question's come up with metagenome data before, because we've had samples that come from human subjects that, I mean, if they're fecal, you'll still get host cells that get sequenced along with everything else. And then, you have to worry about that. But we have had instances in the past where we always say up front, these are only for research purposes. We can't do anything that's clinical. But sometimes we run into gray areas. Examples that I could think of might include research, but on SARS. So sequencing SARS samples,
Yeah. I mean, for better or for worse, right? I mean, I think the COVID-19 pandemic has thrown a lot of this for a loop. Not the least of which is that in October of 2020, FDA, which had in the past several years really began to ramp up its authority to oversee a bunch of laboratory sequencing under its LDT authority, that was essentially nullified by the Department of Health and Human Services, who said that FDA is no longer allowed to essentially authorize any LDTs, period, unless it's voluntary. Essentially, they've lost their enforcement power to go after LDTs that don't otherwise meet their IVD regs, right? It's a pretty big sea change in the way that things have otherwise been conducted. There is now, at least as far, I mean, kind of depending how you look at it, you talk about a regulatory arbitrage opportunity. There's now essentially no authority in the United States that polices clinical accuracy claims from CLIA labs now. I mean, I just, you don't have to do it, right? If you're getting the calls right, you're getting the calls right. You can say whatever you want to say about the clinical utility of the particular thing that you're sequencing. And so hopefully we can work on this gap, but it's going to require a statutory amendment from Congress now, which FDA law scholars have been saying has otherwise been needed for a long, long time. And SARS has just made this impossible. My sympathies to anyone who has sequencing, especially in the beginning of the pandemic, who had sequencing throughput, but was not otherwise a CLIA certified lab, right? Which is a lot of the sequencing throughput that we have.
Just have 1 minute, so I'll ask a quick question, and it needs more answer, but maybe just a quick thought you have. So I'll telegraph it. Henrietta Lacks not being compensated for her cells, the HeLa cells. George Church selling his genome or putting it for sale as an NFT. Is this the direction that we're going in, cryptocurrency, to allow for provenance and reward for genomes? Quick thoughts?
Sure. So I'm going to push the lax side to one case for a second, because it traffics on a whole other issues that, to be blunt, have very little to do with genomics, including like what the standard for unjust enrichment is and what the standards for medical treatment were at the particular Johns Hopkins clinic that treated Ms. Lax back in the 1950s, right? Maybe late 40s, if I'm getting my dates wrong here. Anyway, On the selling your genome as an NFT, yes, one of the things that I mentioned earlier is that as the uses for genomics are going up, one of the things we're likely to see is genomics for fun and profit. And so this is one of the things otherwise that we're seeing out there. One interesting thing that I think is actually worth exploring, I'm just going to come out and say it right now, like, while I think some of the claims for the utility of blockchain have been overstated, I think that there is some good case to be made for finding ways to attach otherwise large data sets that can themselves be readily transferred, think a 30x BAM file, right, with a digital token of some sort to denote ownership, right? And so it seems like, at least in this one context, it is conceivable that NFTs may otherwise have some utility in this regard. And so kudos again to George for making us think the tough thoughts about what the point of all of this is.
All right, genomics for fun and profit. So we'll end there. So thanks everyone for coming, and thanks Jake for a great talk.
Great. Thank you so much, everyone. Thank you so much for having me. And thank you, Gene.